So for coronavirus DNA testing, we have test patient sample 1, and we start off with their cell membrane, their DNA genome, and their mRNA as well. So first off, the genome and the mRNA get removed and the cell membrane is then gone. So DNA must be broken down by an enzyme called DNase, which then goes there. This leaves the mRNA only. So then these two get taken away as it breaks it down, and a primer is added to the A side so that the reverse transcriptase can copy the RNA. Then we have reverse transcriptase making a DNA copy of the RNA, which is called cDNA. The enzyme RNase digests the original mRNA, leaving just the cDNA after that. This is done so that the PCR reaction is allowed to happen with RNase. The virus then has a primer that is added and will anneal if there's a complementary sequence of the cDNA. The Taq DNA polymers can only make a DNA copy of the cDNA if the primer is bound and if it lines up. So for test patient sample 2, we start back from the beginning, but this time we have a virus particle. So the cell membrane is then removed, which leaves just the genome, the mRNA, and the virus. The DNA is digested by the enzyme DNase, which this then becomes the RNA genome, which with this being digested, it leaves just the patient mRNA and the virus RNA genome. The primer is then added to the A's. So reverse transcriptase can make a copy of the DNA, can make a DNA copy of the RNA, which then switches over to this, where the reverse transcriptase is going to make the copy, which is a cDNA. RNase digests the original RNA, which leaves the cDNA and the viral cDNA only. Then we have a primer 2 that is added, which it then attaches to the virus cDNA. The Taq DNA polymers makes a complementary DNA copy of the cDNA if the primer is bound, which it is to the virus. Which is right here, as this copied it. The Taq polymers makes double-stranded copies of the virus DNA, and fluorescent dye shows this happening in a real time. So then we have our Taq polymers with our fluorescent dyes in each of them to show them all.